Good day, this is Professor Will, CBMD PhD. Today is Thursday, June 18th, 2020. It is 8:36 and 46 seconds a.m. Eastern Standard Time, and this is a continuation of my Corona series. So let's get started here. Super potent human antibodies protect against COVID-19 in animal tests. Scientists isolate powerful coronavirus neutralizing antibodies from COVID-19 patients in successfully tested animals. Date June 15, 2020. Source Scripps Research Institute. Summary. Researchers have discovered antibodies in the blood of recovered COVID-19 patients that provide powerful protection against SARS-CoV-2, the coronavirus that causes the disease, when tested in animals and human cell cultures. The research published today in Science offers a paradigm of swift reaction to an emergent and deadly viral pandemic and sets the stage for clinical trials and additional tests of the antibodies, which are now being produced as potential treatments and preventives for COVID-19. The discovery of these very potent antibodies represents an extremely rapid response to a totally new pathogen. In principle, injections of such antibodies could be given to patients in the early stage of COVID-19 to reduce the level of virus and protect against severe disease. Antibodies also may be used to provide temporary vaccine-like protection against SARS-CoV-2 infection. For healthcare workers, elderly people and others who respond poorly to traditional vaccines or are suspected as a recent exposure to the coronavirus. An approach that worked for other deadly viruses, developing a treatment or a vaccine for severe COVID-19 is currently the world's top public health priority. Globally, almost 8 million people have tested positive for SARS-CoV-2 infection, and more than 400,000 have died of severe COVID-19. The daily toll of new infections is still rising. One approach to new viral threats is to identify in the blood of recovering patients antibodies that neutralize the virus's ability to infect cells. These antibodies can then be mass-produced using biotech methods as a treatment that blocks severe disease and as a vaccine-like preventive that circulates in the blood for several weeks to protect against infection. This approach already has been demonstrated successfully against Ebola virus and pneumonia-causing respiratory syncytial virus, commonly known as RSV. Potent patient antibodies block the virus. In parallel, scientists at Scripps Research and IAVI developed test cells that express ACE2, the receptor that SARS-CoV-2 uses to get into human cells. In a set of initial experiments, the team tested whether antibodies containing blood from the patients could bind to the virus and strongly block it from infecting the test cell. Scientists were able to isolate more than 1,000 distinct antibody-producing immune cells, called B cells, each of which produced a distinct anti-SARS-CoV-2 antibody. The team obtained the antibody gene sequences from these B cells so that they could produce the antibodies in the laboratory. By screening these antibodies individually, the team identified several that, even in tiny quantities, could block the virus in test cells, and one that could also protect hamsters against heavy viral exposure. All of this work, including the development of cell and animal infection models, and studies to discover where the antibodies of interest bind the virus, was completed in less than 7 weeks. We leveraged our institution's decades of expertise in antibody isolation and quickly pivoted our focus to SARS-CoV-2 to identify these highly potent antibodies. If further safety tests in animals and clinical trials in people go well, then conceivably the antibodies could be used in clinical settings as early as next January, the researchers say. We intend to make them available to those who need them most, including people in low and middle income countries. In the course of their attempts to isolate anti-SARS-CoV-2 antibodies from the COVID-19 patients, the researchers found one that can also neutralize SARS-CoV, the related coronavirus that caused the 2002-2004 outbreak of severe acute respiratory syndrome SARS in Asia. That discovery gives us hope that we will eventually find broadly neutralizing antibodies that provide at least partial protection against all or most SARS coronaviruses, which should be useful if another one jumps to humans. Rapid isolation of potent SARS-CoV-2 neutralizing antibodies and protection in a small animal model. Hurricane season combined with COVID-19 pandemic could create perfect storm. Date June 17, 2020. Source University of Central Florida. Summary. When extreme climate conditions interact with stressors to social systems, such as the COVID-19 pandemic, the consequences could be severe unless experts from diverse backgrounds work together to develop comprehensive solutions to combat their negative impacts. In the Perspective article, researchers' input mainly focused on the impacts of connected extremes on the water sector. Researchers have extensively worked on many different projects focused on compound flooding, when, for example, storm surges coincide with extreme rainfall or high river discharge. The article brought together scientists and stakeholder representatives with different backgrounds, ranging from the natural sciences to social sciences, public health, and engineering. The authors focus on four main sectors: food, water, health, and infrastructure, where connected extremes often lead to unforeseen impacts. Examples of connected extremes include the impact of Hurricane Maria in 2017 on Puerto Rico's under-maintained infrastructure, limited budget, and aging population, and the spring 2011 Mississippi River floods, in which water was released to protect urban areas at the detriment of agricultural land. A present example could be the COVID-19 pandemic and the current hurricane season. For example, shelters cannot operate at full capacity. Healthcare systems are already under pressure, and emergency funds are depleted. The researchers say many of the most impactful natural hazards experienced over the past decade can be considered connected extremes, where either different factors in the physical climate system combined in unfortunate ways, or the impacts were made worse by interactions between physical and societal systems. It is important to recognize and treat connected extremes as such, and for scientists from different fields to engage directly with stakeholders and decision makers to develop new, robust, and flexible policies to better combat their negative impacts. A coordinated COVID-19 response helped Western Washington State flatten the curve. Date June 16, 2020. Source: American College of Surgeons. Summary: a regional coalition guided multifaceted approach engaged health systems, long-term care facilities, state and local governments, and organizations to rapidly respond to the outbreak. Despite having the first confirmed case of coronavirus and the first major COVID-19 outbreak in the United States, the state of Washington implemented a response plan that kept its death rate the lowest among all states that have had major outbreaks. A multidisciplinary consensus panel of 26 experts analyzed Western Washington response and identified 6 key factors that contributed to flattening the curve in the state. 6 pillars of the COVID-19 response identified by the consensus panel are early communication and coordination among stakeholders, regional coordination, and situation awareness of the healthcare system, rapid development and access to testing, proactive management of long-term care facilities and vulnerable populations, and effective physical distancing in the community. As of June 5th, Washington State had 22,729 confirmed cases of COVID-19 and 1,138 ensuing deaths, a rate of about 5%. The overall death rate in the United States is 5.7% according to Centers for Disease Control and Prevention data. New York State, by comparison, has had more than 380,000 confirmed cases and 30,000 deaths, a death rate of 8%. Along with the governor's stay-at-home and physical distancing orders, pre-existing relationships across the healthcare system were critical in facilitating this response. The consensus panel noted that early communication and coordination among the various hospital systems, emergency medical services, LTFCs, was pivotal in the response. Normally we exist in a competitive landscape, but there was great willingness for each system and entity to do what they could to address the region's needs. Another key component of the response was the ability of the University of Washington Virology Laboratory to rapidly develop viral testing and make it widely available. They recognized very early that there would be a need for widespread testing that would likely exceed the capability of the state laboratory. They worked to develop their own testing platform and validate the test so that they could begin receiving samples from the initial outbreak. As of June 5th, 383,587 people have been tested for SARS-CoV-2 in Washington. Researchers say on-site support includes COVID-19 testing for residents and staff, and training for personal protective equipment and effective isolation and quarantine. Researchers were able to keep many of these patients in the nursing facilities and not overwhelm the hospitals by evacuating entire facilities. In addition to people in the LTFCs, the response identified other vulnerable populations, minority and immigrant communities, and non-English speakers, the homeless, and people in jails and prisons. PHSKC has convened 9 task forces and community advisory groups to engage diverse populations. To service the homeless, King County added shelter space, hand washing stations, and call center, along with locations where they can access isolation and quarantine services. Each region should begin to organize itself in this way, because whether it is a pandemic or other type of large-scale disaster that occurs, it is critical for regions to prepare to develop communication structures that support situational awareness and patient distribution strategies. Otherwise, facilities get overwhelmed and patients suffer. The authors propose these lessons learned as a roadmap for preparation for future outbreaks, as well as establishing a lasting infrastructure, which will strengthen the healthcare system's response to all future mass casualty events and save lives. Good day, this is Professor Will, CBMD PhD on infectious disease, molecular microbiology, and pathophysiology. Well, hope you enjoy listening. Until next time, Have a great day.